Alright, so welcome to this video on chromatography, an essential analytical technique used widely in chemistry, biology, and medicine. Today, we're going to explore the various types of chromatography, understand their principles, and learn how they are applied in medical and biotech fields. Chromatography is a powerful tool used to separate and identify compounds from a complex mixture. The underlying principle is pretty straightforward. Different compounds in a mixture will interact with a stationary phase and a mobile phase in distinct ways, causing them to move through the stationary phase at different speeds. This difference in movement is what allows us to separate and analyze the individual components. The process begins by placing a sample onto a stationary phase, typically a solid medium. The mobile phase, usually a liquid or gas, passes through the stationary phase, carrying the components of the mixture along with it. Depending on their affinity to the stationary phase, some compounds will move more slowly than others, leading to separation. This process is called partitioning. There are several types of chromatography, each with its own specific applications and advantages. So let's start with thin layer chromatography, or TLC, and paper chromatography. Thin layer chromatography uses a thin layer of silica gel or alumina as the stationary phase, while paper chromatography uses paper, typically made of cellulose. The sample is spotted onto the stationary phase and then placed upright in a solvent. The solvent moves up the plate or paper by capillary action, carrying the components of the sample with it. More nonpolar substances will travel further up, while polar substances will remain lower, adhering to the stationary phase. We can calculate the retardation factor, RF, for each substance, which helps in identifying unknown compounds. The RF value is calculated by dividing the distance traveled by the substance by the distance traveled by the solvent front. Column chromatography operates on the same principles as TLC, but uses a vertical column filled with silica or alumina beads. The mobile phase flows down the column due to gravity, separating compounds based on their interactions with the stationary phase. The compounds are collected in fractions as they elute from the column, which is particularly useful for purifying large quantities of a substance. Ion exchange chromatography uses a column with charged beads that attract oppositely charged molecules. This technique is excellent for separating proteins or nucleic acids based on their charge. A salt gradient is often used to elute the bound substances from the column. Size exclusion chromatography relies on the physical size of molecules. The column contains beads with tiny pores that small molecules can enter, slowing them down. Larger molecules bypass these pores and elute faster. This technique is frequently used to separate proteins and other macromolecules. Affinity chromatography takes advantage of specific interactions between a molecule and a ligand attached to the stationary phase. For example, a column can be designed with antibodies that bind to a particular protein. After the protein is retained, it can be eluted by adding a solution with a higher affinity ligand. Gas chromatography is unique because it uses a gaseous mobile phase, usually helium or nitrogen. The sample is vaporized and travels through a coiled column filled with a solid or liquid stationary phase. Compounds are separated based on their boiling points and affinity to the stationary phase, and are detected as they exit the column. Finally, high-performance liquid chromatography, or HPLC, is similar to column chromatography, but uses high pressure to push the solvent through a tightly packed column, allowing for faster and more precise separation. Modern HPLC systems are computer-controlled, allowing for precise gradients and temperature control to optimize separation. Chromatography isn't just a lab technique, it's a vital tool in medicine, health tech, and biotech. In medicine, chromatography is crucial for analyzing blood samples and detecting various biomarkers. For example, GCMS is often used to detect metabolites and drugs in toxicology screens, providing critical information for diagnosing and monitoring patients. In the biotech industry, chromatography is essential for purifying proteins and nucleic acids, which are foundational steps in developing vaccines, monoclonal antibodies, and other biopharmaceuticals. Affinity chromatography, in particular, allows for the high purity isolation of target proteins by exploiting specific interactions, such as those between antigens and antibodies. Chromatography is also a cornerstone in drug development and quality control. HPLC is routinely used to ensure the purity and stability of pharmaceutical compounds, directly impacting drug safety and efficacy. So, to summarize, chromatography is an indispensable tool in both the scientific and medical communities. From TLC to HPLC, each technique offers unique advantages for separating and analyzing compounds. Understanding these methods is crucial for anyone working in chemistry, biology, or medicine. And if you found this video helpful, don't forget to like, comment, and subscribe for more insights into these different techniques. And if you're watching this on our app, please make sure to leave a like there and comment on what topics you'd like to see next. Thank you for watching, and good luck studying.